I think I'm online again. Let me quickly. Yes, everything seems to be working, and sometimes I'm a little bit surprised how quickly a week goes by. Hi, hello, and welcome again to another Saturday Microscopy live stream. I see that many of you already joined in. Let me do a little bit of housekeeping first to see if everything works. It's always a yes. Okay, we've got 28 people online right now. Cool. Let's get started. First of all, again, hello all around the world. I'm Oliver. Let's do a little bit of sample preparation and microscopy again today. I see that many people have already joined in. There is actually someone from Ireland. Hello, Oregon, Atlanta, Lebanon, Vietnam, Los Angeles, soggy UK. It's been actually snowing here in Central Europe, in Austria, where I am. And now basically the snow is gone again. From France, from England, California, Connecticut, the United States, Poland, New Jersey. Nice to have you here again on the Saturday. I will be honest with you, I actually planned to do something different today. I actually wanted to do a little bit of a pollen, to grow some pollen tubes. If you've joined in last week, remember that I had a flower, and these are the anthers with the pollen of the flower. And I wanted to actually make them germinate so that they will form a so-called a pollen tube. And I prepared everything and mixed everything. It did not work. So I'm still working on it and trying to figure it out. So I had nothing to show you, and that's why in the last second I decided, okay, doesn't matter. We're going to put a little bit of I'm going to show you a DNA extraction activity. A few years ago, I think I've already done that, but every time it's a little bit different. So I would like to extract my own DNA, and we're also going to put it under the microscope, okay? And I've got a banana here, and even some, and I just tried it 10 minutes before the start of the live stream. I tried it with some fruit juice. Okay. However, and it did work. I did get a little bit of DNA out, but a very small amount only. So this is not the ideal thing. But I want to show you in any case what maybe the banana should work quite well. So I see that more people joined in. Okay, I forgot where I stopped saying hello, California, Connecticut, Poland, New Jersey, Jerusalem. Okay, very nice. So I want to give you a little bit of theory first, and then later on we're going to see how far we're able to get. And otherwise, I'm just going to show you a few other things. You see what I've done here. That's the flower from last week. Right, I stained it a little bit so we can do again some cross sections of the plant stem. This time stained. We're going to see how far we're able to get today, and then we're going to take it from there. Okay, from the Maldives. Wow, that's nice. I've been also many years ago at the Maldives already. So I'm going to quickly tell you the theory first, and then I'm going to do the experiment, so to say. I would like to start off by actually getting my cheek cells out into a little beaker here, okay? And I do that by flushing my mouth with salt water. Excuse me, now I have to cough. And the idea is that those cells that are flushed off, I want to break them open using simply some soap, some dishwashing soap. And then I'm going to add some alcohol to precipitate the DNA. And then we want to put the DNA under the microscope. Okay, so it's just a couple of steps. It doesn't take long, just a couple of minutes. And I'm going to basically comment on that. It's almost what I would say a foolproof experiment. So far, it always worked out. It's kind of not every experiment is like that, but so far with me it always worked out. The only question is how much DNA are we going to get, and I also need to tune down a little bit the expectations. When we put it under the microscope, don't expect some super fancy, cool looking thing, okay? If we're able to see something that is string-like, then I'm already happy. And yes, I need to explain why it's even possible to see the DNA under the microscope, because actually it's way too thin, okay? Do you think I've used this? There is a reason why we use salt water. And the reason is because salt water is more polar. And what I need to do later on is I need to add alcohol over the suspension, and therefore the alcohol and the salt water, they don't mix quite as well. That's the reason we have to do that. Okay. So what I have prepared here is quite simply put. I'm going to put a little bit more water over there, maybe. Yeah. Look, this is just some regular tap water, and this is just some regular salt. And all I'm going to do is I'm going to add a little bit of salt in here. I'm not. It's more than enough. That's the amount is not really critical at all. Okay, and I've basically decided to prepare as little as possible. So I want to show you as many steps as possible. And while I prepare this, I can talk about a few other things. Okay, so yeah, let's talk about those. The DNA again. The reason why we need to flush it, of course, is to get the cells, and the dishwashing detergent is there simply to break the cells open, because it's soap and the cell membranes are basically lipids, phospholipids as well, and therefore it breaks open the cell membranes and it releases the DNA. Okay, the DNA is not pure, right? It contains a lot of proteins and impurities as well. And that can actually be an advantage, because sometimes these impurities that are sticking to the DNA actually make it more easily visible. Okay, yeah. For those of you who've just joined in, okay, I'm trying to do a little bit of a DNA extraction. DNA is atoms connected in sequences. You can see them. That is correct. The DNA is way too thin to be seen directly under the microscope. But why we're still able to see it is it because maybe thousands of DNA's molecules, when they kind of stick together, and when there are proteins and other impurities sticking to it, we can actually see the string-like consistency, and that's kind of the idea. So individual DNA molecules are way too thin. You cannot see them, obviously, right? Yeah, you see it's kind of still not completely dissolved. Maybe I've added a little bit too much salt, and there's of course always a danger if you add too much salt, then there's this reflex that you have to throw up, so you have to be careful. Yeah. So what I'm going to do now is I think that's going to be fine. You see the salt here, collected on the bottom. So I'm going to take a small sip, and for about a minute or so, I'm going to rinse my mouth, and I'm going to spit everything into this beaker in here. Okay. So that's kind of the idea. Yeah? For your information, this method of extracting DNA, that is basically a very similar method is also used, of course, in laboratories to extract DNA out of a bacteria, for example. So what I have done many years ago, I did some DNA analysis on bacteria, and what we did is we did not break it open with the dishwashing solution, but we broke it open by quickly freezing the cells, and by freezing it, the water expands, and the expanding water breaks open the cells. We used actually liquid nitrogen to do that. Yeah. Can you swish plain water and spit it out and then add the salt? I see no reason why this should not work. I think that works, right? If you just use plain water and then add the salt later, I see no. Yeah, it's not gonna taste good. Cheers. Yeah, as you can see. Yeah. Oh my gosh. You know what I'm gonna do? There's even some food particles in here. You know what I'm gonna do? I'm gonna do this again. Nah, maybe I'm not gonna do it again, because I already rinsed off many of the cells, and maybe I'm not gonna get enough cells. So what I'm going to do therefore is I'm just gonna leave it, because the food particles and whatever, they have settled down here on the bottom, right? And now the next, the next thing what I'm going to do is now I'm going to add a little bit of dishwashing soap. So that's basically just regular soap, nothing special. It's clear. And how much should I add? I don't know. Instructions say a drop or so. And what is very important now, and that's the first, I'm gonna just add four drops. it's 200 microliters. I just need to say the following. What is now really important is after you've added it, and I can tell you because I tried it out, okay? You have to stir carefully. If you really mix it too aggressively, what's going to happen then is that the DNA that's released from the cells is actually going to rip, and we don't want that. So the instructions are to carefully, let's do it like, you know what, I'm going to maybe move this over a little bit like this, the camera. Here, that's better, yeah? What you have to do is you have to carefully do it like this. I've once experimentally really shaken it very aggressively because I simply wanted to test it out and I could not get any DNA out. Okay, yeah, so that is just a important thing, and we're just gonna. If you shake it, of course, too much, then there is the danger, not the danger, but the possibility of foam forming. Yeah. So I'm just I'm going to read also some of the comments here. Let's hope we don't see any rotifers in my mouth. Yep. There are plenty of bacteria. I mean, saliva is fascinating because there's so much stuff in there. I mean, I tell you, I've done this already several times before. So far, it always worked right away. And maybe this is going to be the first time where it doesn't work. I don't know. Okay. And we're going to see. Yeah, I think it might be enough. So what I'm going to do next is the following. I need to take a little bit of alcohol. It is just regular ethanol. Cheap ethanol, and ideally the ethanol should be ice cold. It's not in my case. And what the alcohol does now is the following. I'm going to tell you what we have to do. Is we're not allowed to mix it. What we have to do is we have to carefully pour the alcohol over this here, so that there is going to be a layer of alcohol on top. Okay, so that's the idea, right? I show it to you again over here. Yeah, I have to carefully pour it over and tilt it and let the alcohol run on the side of the beaker because I do not want it to mix too much, right? And then there should be a layer of clear alcohol here over the surface. Yeah. Can you use isopropyl alcohol? I've never tried it at the university. So we always used regular ethanol. I see no reason why it shouldn't. No, well, I don't know. I mean, regular alcohol dissolves better with water, and it has to dissolve a little bit. I would simply try it out, but I think isopropanol is more expensive. Okay, yeah. I'm curious, would this method work with spermatozoa cells too? I think so, yes. And I think hypothesis it might work even better because, to my knowledge, the spermatozoa are kind of very fragile. So if you add some detergent, they should break open more easily. However, yeah, never tried it. So let's do the following. I'm going to. How am I going to do this? Yeah, I'm going to do it like this, maybe. And I'm going to do it like this, and now this is where it becomes you need a steady hand. I need to pour the alcohol carefully on the side, so that a layer of alcohol forms on top. So you need to be very patient. Maybe this is enough. Okay, and let me quickly have a look. Yep, we are very successful. Okay, yes, it works. So the live stream is saved. It's also now worried that things don't work out. So I'm going to show you now over here what we see. I have to tilt the camera. I can't even focus it closely. Let's put it away a little bit further. Yeah, that's basically my desk here, okay? Yeah, and let's see if we're able to focus. Yep. Here we go. So this is very nice, actually. I'm going to try. Yeah, no, I need something to point to. Okay, so down here down here, this is of course the dishwashing solution with the salt water, which are flushed out. On top here, the clear liquid over here on the top that you see, that is the alcohol. And then you see those bubbles here, right? Yeah, and this basically, that is the DNA. So because the DNA does not dissolve in alcohol very well, it starts to precipitate out, right? So basically, and those bubbles here are because any dissolved air or oxygen in the water, it kind of bubbles out. Okay, because it doesn't dissolve in alcohol very well. So, and now, if we wait a little bit, the bubbles actually will do the following: they will rise up and they will start to pull the DNA out. So those bubbles here are caught in the DNA. Yeah. Yeah. So and that's actually, you have to tell you, this is actually quite a lot of DNA, right? And if we wait a little bit and if we carefully mix it more, so that the alcohol and the water over here mix a little bit, then more and more DNA will start to precipitate out. Yeah. And what we can do now is the following: I'm going to take some toothpicks, and you might be actually able to see that it starts to pull strings when we go in with a toothpick. Do you actually see this? Yeah? Did you just see this a little bit? Maybe let's turn this around here. Yeah? I don't know if you're able to see this. It's very tiny. It starts to actually pull strings. Yep. You have to look at the top. Yeah, and it looks a little bit slimy, but it's pulling. Yeah, here, those strings. Yeah, but the very top. Yeah. And what you can actually do, and this is what we have done, is we've done the following. We've actually wound up the DNA on toothpicks. Here we go. That's actually a nice view here. Yeah. Obviously, this is probably millions of DNA strings sticking together, right? And we're able to pull. Yeah. This works quite well. I'm going to try to put this carefully on a microscope slide. Now, don't have too high expectations, okay? Don't have too high expectations. You're definitely not going to see a double helix or anything like that. But sometimes, if you're very lucky, you can actually see that there is a, how do you say, that you can actually see the strings. No, I don't know. I'm going to just give it a try like this. I'm going to now add a cover glass. Yeah, it looks more slimy than anything else, but still, I think it's kind of fascinating. I don't know. It's not very. Yeah, a little bit of liquid in here. Let me quickly check if it looks okay or not. And where is the scope view? Up. Oh, yeah. Lots of other stuff here as well. You see that's yeah, very thick. Yeah, but you see it's contains probably thousands of DNA molecules. Yeah, sticking together with a lot of impurities and proteins and whatnot. Yeah. I've actually seen nicer ones than this one here. Yeah, look, some fiber. Yeah, maybe this was some contaminant that was already on the on the slide. Yeah. Yeah. So this is basically let's try another one, okay? But I just wanted to show you a little bit how this actually works. And maybe, I don't know, let's try the following. I'm going to try now something totally crazy. I'm trying to try a different method, a different technique. Turn off the lights here. And with a banana, we should actually get even more. Maybe by going in here like this. I don't know if this is somehow possible. No, it didn't catch anything. I don't know if I got anything here. Let's quickly put it under the microscope as well without a cover glass. Okay. I mean, I once had the this is yeah, here. That's better. It's starting to dry up a little bit. But do you see those very fine strings here on top? Yeah. Where are the arrows? Yeah. Here's the arrow. Where's the arrow? Just let me see over here. See those fine strings over here, right? Yeah, you see, these are also not individual DNA molecules, obviously, but also many of them stuck together. Yeah. Now without cover glass, yeah, let's go up yet higher. Okay. And it starts to dry out, and yeah. Here we see that it's very stringy. Yeah. So that's kind of the thing that I kind of wanted to show you. And let's see, as the alcohol, of course, evaporates, maybe we're not able to see it quite as well anymore. You know what? I'm going to try it very quickly. Let's see if we're able to. That's phase contrast. Okay. Let's do the following. Let's open it up. Here, that is phase contrast. Yes, and here it's a little bit better. Yeah. Yes, there's lots of. The question is how do you know that it's actually DNA? Because there's lots of biological material other than DNA in the cell. That's correct, but there are not so many of them that are actually that long. Number one. And number two, I know it must be DNA because the way that when we wound it up, that's actually the way that we're using to actually do DNA analysis. Yeah. So what I've done is we've then taken out those string-like structures. Yeah, and we've then purified it. We added phenol to remove the proteins, for example, and then we dissolved it again, and we did several purification steps in order to do a so-called DNA analysis. Yeah, so this is actually a standard technique that is used in labs. Right, but what you see over here again, I know I'm repeating myself. The DNA is way too thin. You would not be able to see it here. So each one of those strings that you see here is definitely not one DNA, but maybe hundreds or thousands of them. And then there's still impurities and proteins and so on sticking to it. And that's why it's even visible at all. Yeah. So I just wanted to show this to you. Actually, I'm quite happy that at least we're able to see this string-like. Yeah, here still are some cells down here. Yeah, so. Because eukaryotic DNA, like also in humans, is not just pure DNA. It's wrapped around so-called histone proteins. Yeah. And therefore, the whole thing is actually much thicker. Yeah. So this could, and lots of other stuff is also sticking to it. I can imagine that when you added the alcohol, that the proteins started to denature, they changed their shape, and then they started to stick to the DNA, possibly. Yeah. So just that. So let's have a look how this looks in bright field, because most of you have actually bright field. This is not what this is here. I have to close it, turn it down here. Yeah, barely visible. Normally, if the contrast is so bad, what you normally do is you take a picture, a screenshot or a picture, and then you really go into Photoshop, and then you go up, increase the contrast. Yeah, but you see, it's kind of difficult to see. Here, you have to close the condenser all the way. Yeah, and then So okay, I'm just checking if there are any other comments. So far, not. I don't know. We can do the following. I can try to do the whole thing now with a little bit of banana. Okay, let's same principle with a banana, and in this case, I'm not going to use salt. I'm just going to use regular water. Ooh, I nearly tipped over the. So I don't know. This might be a little bit too much water. Desk, and then I can show you. Let me. Quickly, I need to add, just a second, I need to quickly add a little bit more water over there. It started to dry out. I wanted to show you something. Where is it? Just a second, I'm doing something else now, okay? Because I hope that they didn't die, because this was a little bit something I just wanted to show you. Let me quickly check. I'm just doing something different right now, okay? Because I prepared something else and the slide actually dried up. They're still okay. Yeah. Still kind of okay. Yeah. Okay. No, I had just for your info, I had I put some sea monkeys under the on a slide, and the water started to dry up a little bit, and that was the problem. So we're now going. Yeah. When you want to do a banana, you take a banana, and where is it? And banana has the advantage that the amount of DNA that you're actually able to get out is way more. It's much more. And I'm going to now do the following. The banana will then be eaten later anyway. Maybe so much. That's why you always make sure everything's reasonably clean, so that it can be. And you take a piece of banana, goes in here, bloop. And then all you do is you try to crush it open. Other people have said strawberries also works. Onion also apparently works. And I'm just thinking, did I not do this already a couple of weeks ago? Where I did not do DNA extraction, but I extracted the starch, banana starch. This is also a way of actually having a look at banana starch. And you can increase the contrast in the camera software you're using. This is the question. The thing is the following. I'm not using a camera software. I'm using a program called OBS Studio, and I'm getting directly the live feed from the camera into the... actually, I should be able to increase it a little bit, but then I have to go into the settings. So I'm not using a dedicated microscopy software. So I can adjust the contrast a little bit, but the options are there are not many options to do that. So it's not possible to fine-tune it. Right now what I'm doing is I have got, of course, several cameras, and they all go into my computer, into this program called OBS Studio, which is a free program, which I can highly recommend. And I also use this program simply for, not only for live streaming, but also for recording when I make, generally when I make videos. Yeah. So, yeah, I think this should be enough, huh? The next thing that I want to do is the following. I have something like a little sieve here. Now this is going to be messy. Maybe I'm going to spill something. Yeah, of course I spilled something. But it doesn't matter. Yeah, it's a mess, as expected. But I've got a sufficient... Let's throw this away here. Okay. Wipe everything. I mean, it looks kind of similar to the salt water from my mouth from before. Yeah. If you let this stand now a little bit, then what you're going to see is you're going to see that there's some starch going to settle down. Yeah. So, I mean, honestly, if you want to, we can also have a look at that first. Yeah, but it's kind of banana starch, and this banana starch is something that you might also want to have a look at or not. I mean, as we're waiting for everything to settle down, why not take a small sample? And why not take a small sample? And let's see if there's some banana starch here. The banana starch, I don't want to have this mixed in, so I'm just going to wait a little bit for it to sink. Let's see maybe also some cells, whatever. Yeah, here, exactly. Yeah, these are those things that you see here. These are the starch grains, basically. Yeah, yeah, still cells around it. Yeah, yeah. Okay, so you get the idea, and we now have to do the following. I nearly forgot. I have to, of course, also add again a little bit of dishwashing detergent. Should I try it like this? I don't know. To do that, take some. So this is very. There are different ways how you can break it open. I already mentioned that I've used in the past also yeah, liquid nitrogen, very cold, which when you shock freeze the cells, then the water expands and this breaks open the cells. You can also use for bacteria, if you want to break them open to extract your DNA, you can use enzymes like lysozyme. Yeah, so there are a variety of ways. Yeah. Again, here you don't want to mix too aggressively, otherwise you're breaking the DNA itself. And we're going to let the detergent do its action again for a minute or so. I've seen people simply blend the fruits instead of mashing them. Yes, blending also works, but you have to keep it short, the blending. Usually and indeed, also in the lab, we do this sometimes, and the instructions are you have to blend it in short bursts. Of course, some of the DNA will basically become damaged, but of course, as you blend it, of course, you also break open more of the cells at the same time. Yeah? But once the cells are broken open and you keep on blending it, then you might actually destroy the DNA. Yeah. Okay. Yeah, OBS has a contrast filter. That's correct. And what I have to do is I have to go into the settings and change the contrast and so on. Yeah. So yeah. So I hope that this is gonna work. And what I'm going to do next is I'm going to do the same thing as before. I'm going to, yeah, that's the alcohol. Carefully pour it over. And because the DNA does not dissolve in alcohol very well, it starts to precipitate out. So let's hope that this actually works. So I'm going to show you again. Yep. Look, all of this stuff that's floating here on the top. And as it rises up, maybe you are able to see that there are sometimes strings still going downwards. Now, because it's so concentrated, usually this DNA is not very pure, right? So there's lots of other stuff also sticking in the DNA. Does the DNA size vary depending on the sample? Well, let's put it this way. The amount of DNA definitely does depend on the sample. It depends how many cells are present. So that's definitely I would say the one factor. And the other one is how many of the cells actually break open in the process. I mean, you can have a lot of cells there, but if they don't break open because the cells are very stable, then of course you're not going to get a lot of DNA out, right? And yeah, so let's have another look. Okay. Yeah, this one here. Honestly, I've already had better. Samples. Yeah, it did precipitate out here, but it sticks together quite a bit. But if you see over here, if I pull over here, then a little bit, yeah, afterwards it's dragging something along. Yeah, and And what you usually do is you try to go up and you try to go down and dip it into the solution, and this way you're carefully mixing it. And then you're able to hopefully get... this is not a lot what I've got here. It's kind of interesting. Sometimes this works really well. And sometimes... I mean, I've got plenty of stuff here, but somehow the consistency is not quite the way that I hoped it to be. Okay. Or I will try to do it like this. Let's see. I have some doubts whether actually there is something on here. Nothing. Oh, okay. I think sometimes it slips from the slide. Because if there's still some DNA hanging into the liquid, then sometimes it slips off again. I think this happened here. Okay, we gotta try again. Oh, there seems to be something here a little bit on the side. Let's try this here first. No, that's nothing. Nope. Yeah. Otherwise, I'd have to try it again. But I mean, you get the idea. And what time is it? I need... yeah, okay. Need to quickly go back to the desk view. No, this is interesting because I used to get much nicer banana samples. The camera is not quite focusing either. Sometimes it helps to dip it in. And if you keep on doing that over the course of maybe 15 minutes, 20 minutes, you can actually wind up quite a bit of DNA. There seems to be something on here. See if I can get it off. Okay, if this doesn't work, then I'm just going to leave it because luckily we've seen it already before. Because the DNA sometimes slips off. But you see over here, yeah, there is, look. It's very string-like. So I'm scratching it off now. I don't know, this is not a lot. I think I might not have gotten a lot on here. Nope, it slipped off. It tends to stick to the, yeah, some banana cells, of course. Nope, this one is actually I declare this officially as a failure. Okay, which surprises me because normally this works quite well. What I can try, however, is the following. I'm going to try this again. I'm going to put this over. I'm going to dump this in here. And I'm going to now try to take some of the unfiltered one. I'm going to add again some oh, you don't see this. I forgot to switch over to the desk view. So I've taken, so that's gonna be my last try. If it doesn't work, doesn't matter. And I'll show you. Okay, the others are fine. It could also be that you have to let it sit a little bit longer with the detergent. So, and so I would like to, simply the entire human genome stored in the human cell equates to 3 to 6 gigabytes of uncompressed data. For the same size of transistor NAND flash storage, you would get approximately 40 bytes. There is actually, this is some people have been also even considering to use DNA as a data storage to encode information on the DNA. This is also quite interesting. Yeah. Back to the drawing board. Faulty banana. Yes, we declare it's the fault of the banana. How is that? No, this is actually just for your info. This is when you're doing a lab work. This is actually something very common that I would say a lot of the experiments, for whatever reasons, they don't work out quite the way that we expect them to work out. Yeah. So maybe I should have crushed it a little bit more. Yeah. Let me see. Yeah, but it is this stuff precipitating, no question. So let's have a look again. So does this not work? I already see I probably need some kind of a macro camera. Or maybe because it's... Here we go. Yep. Okay. Yeah, this one actually is interesting. It does not look quite as it looks like it's flocculating more than it's actually string-like. See, but if you pull over here, I mean, the rest also pulls along, so it is somehow connected. Let's try to wrap it up a little bit. I mean, there are lots of banana cells in here as well. Here we go, at least there's something on here. Okay, I don't know. I don't know how good this actually works, but it's a little bit just a proof of concept. And afterwards, I shall move on a little bit. So, let's see. This is more slimy than anything else. It's mostly banana cells. But you know what? Let's go again. Phase contrast. So now we have to ignore the banana. Yeah, too many banana cells. So filtering should have been, Nope. I was actually putting so much hope into the banana. I was actually expecting the one from the mouth not to work that well, and the banana better because there is actually there are more cells. But I think what I should have done is the following: maybe used indeed a blender. Maybe this would have actually ripped open the cells more. It could be that the detergent, because this is also something that I've done before. We've used some blender. Yeah. The cells were not lysed enough. That's correct. Yeah. Centrifuging would allow me to separate indeed the cells from the supernatant. But I think the problem that we've got here is you see over here on the outside always the cell wall, and the banana cells were clearly too strong here. Don't forget one thing. That might be the explanation. That my cheek cells they don't have a cell wall. Right, so indeed, maybe it has to be a little bit more aggressive, or I tried the enzyme cellulase, which also digests cell walls. Okay, so take-home message is the following. For plant cells, we need to blend it. Okay, not too long. Otherwise, that's phase contrast, not phase. That's dark field. Yeah, so that's the thing. Evidently, simply crushing it, the bananas won't work. Okay, yeah. So that's a little bit of a pity, but at least we've seen it with my cheek cells before. So what I'm going to do now is I'm going to do a little bit of cleanup now, because it's getting very messy here. And yeah, I want to move on. And the next thing I want to show you is, and maybe some of you have already seen it, a couple of days ago I made a video with Artemia. These are brine shrimp. And I would like to show you what I've got here. Okay. And yeah, I've got over here, this is a slide where I made a ring in here, simply to hold the water droplet in here. And what I've got in here is, I think there are two tiny, what they call so-called sea monkeys. They used to sell them for kids. And I've basically also tried to breed them over here in this container here. And it kind of didn't work very well. It worked better a few years ago. But I think that the eggs are already kind of old. But I just want to show them to you. Because, and here they are. It's now in dark field again. And those dark circles that you see, these are eggs, right? Yeah, and this is how they're basically moving around now. You see this one. One of them is smaller. It's a little bit younger. The other one is a little bit older. And on the outside here, that's where I've got the ring. Yeah. Yeah, so there is a question here, what makes phase contrast unique? Well, phase contrast, for the invention of phase contrast, the guy Fritz Zernike, he actually got the Nobel Prize in physics back in the day, actually was one of those techniques that somewhat, I would say, revolutionized light microscopy, because phase contrast allows you to see transparent objects that are otherwise impossible to see, and the optics converts it into differences in brightness. So you're able to see bacteria, for example, much better without staining. So you're able to observe live cells, and you're able to see them, because otherwise they would be transparent, right? And so phase contrast in that sense. However, phase contrast, I also have to say, does not always give you very nice looking images. So the specimens have to be very, very thin. Yeah. Yes. Yeah. So basically, when they hatch, they carry along some yolk for food, and yeah. So not sure if you saw my comment yet on the Artemia video. No, I did not see the comment. Yeah, no, because I made just a couple of days ago, I made a video. And what I want to do now is the following, and this is also something I did not do yet, is I want to slow them down. This here is a solution of 2% nitrocellulose, which is, yeah, very viscous solution. And what I would like to do now is I would like to add that to, yeah, you know what, we just can also go regular bright field over here. Let's flip this away. Yeah, this is now regular bright field, and because they're kind of moving a little bit too quickly for my taste. Where's the other one? Here it is. Okay, yeah. What I would like to do is I would like to add a little bit. Yeah, I would like to add a little bit of this yeah, nitrocellulose solution, and this basically should slow them down. Yeah. Yeah. Yep. Ah, yeah. So 3 months. I don't know. 3 months. That's a lot. I was not able to keep them that long, and those sea monkey eggs are sold. Yeah, not only for kids, but actually for people who have a home aquarium, because people will actually breed them as for live fish food. right? So you buy those eggs and you grow your own fish food, so to say, right? And because it's moving a little too quickly, what I would like to do now is I would like to add a small amount of this solution over here, which is very thick. I'd like to add this and hope that this kind of slows them down a little bit. And in order to do this, I'm going to go back to the desk view. Okay. And yeah. The dark spot thing that you see in the center, these are the eggs. And let's add a little bit of this. And I wonder if this actually works. You know, it has to be suspended a little bit, lots of bubbles now. Let's have a quick look. I hope that the bubbles pop. Maybe not enough. This one here is already a little bit slower. Yeah, because they're not able to move quite as quickly in this nitrocellulose solution because it's very thick, very viscous. What about the other one? Yeah. Maybe you have to add a little bit more. Yeah, so to slow it down yet further. It's a lot of work for them now to swim, yeah? Yeah, if you float a cover glass on top of them, then slowly take water away till they are held in place but not crushed. Okay, that's a possibility. You basically, what I have right now is I've got this ring around it, so I might also be able to drop a cover glass directly on on the surface of here because it acts like a spacer. But I'm going to add still a little bit more of this. So I hope that they're still in here. So a toothpick to pop the air bubbles. Okay, and let's hope for the best. Yep, here we go. And that's one of them. Is the other one still around somewhere? Or did I here's the other one. Okay, yep. Yay, yeah, that's, yeah, indeed. It's much easier to observe them now, yeah. I'm going to now do the following. I will go up with them. So again, I'm working without cover glass here, okay. Yeah, and yeah, you can even see the digestive system. Would be interesting to see actually if when they eat food, whether the food is able to move through the digestive system. Yep. And the other one must be over here. Bumped it. So, yeah. Yes, they seem to be still quite young. I've also had much larger ones. They already look like tiny shrimp. Okay. Yeah. I just wanted to also show you a little bit the where I've grown them over here. I'm just going to quickly for a minute or so just show this to you over here. Yeah. So those dots that you see down here, these are the eggs. And it was a mixture of salt and those eggs. And then if you take the correct amount, then essentially, yeah, it will also adjust the salt concentration of the water at the same time. But there are thousands of these tiny eggs, but not that many actually hatched. Yeah, so maybe there are only, I don't know, 10, 15 or 20 of those actually hatched. So it actually shows that the culture itself or the eggs are not that good anymore. Yeah. What do they eat? A good question. I would say probably tiny suspended particles. I can imagine maybe some microscopic algae and so on. And it actually comes in the pack. There was already also some food included. Yeah, and it's also possible to buy food for them in a liquid, and you have to add a small drop of this. And I suppose that these are some kind of algae. Yeah. They're pretty yeah, salt tolerant. So if you look at the pack, you're gonna see that yeah, there's half of the contents are eggs, half of the contents are is actually just salt. Yeah, so they're able to withstand pretty high salt concentrations. You're not able to see them swimming around here quite well. Yeah, but if you want, we can quickly have a look also under the stereo microscope. Okay. You can actually see some of them, yeah, swim around. But the contrast is very, very low. Oh, where is this? Yeah, over here, for example, this one swimming here. Yeah, but you see, yeah, down here, the contrast over here, there is one. Yeah, I have to probably refocus a little bit because they're of course higher up in the water. Yeah. But you see, again, compared to the high number of eggs, only very success rate was quite low. Yeah. But in any case, fun to observe. And what I would like to do. Yeah, these are yes, these are Artemia eggs. That's correct. Just a second. If you're interested, I'll just show you. Just a second. Okay. Yeah, they basically came in a box like this for kids, right? But the actually interesting thing is this pack in here, because this is for sold as fish food. So you have to grow it yourself, and if you look into it, then you can see that. Again, I have to switch now to the desk view again. Yeah, you can see that it's the white stuff that's salt. Yeah, mixed with eggs. Yeah, and you take a. It says you have to take a tiny spoonful and mix it with 20 milliliters of water, and then the salt concentration is also automatically adjusted. Yeah. Sprinkle on toast for extra protein. Yes. Yeah. So this is kind of the thing here. So yeah, kind of a fun project as well if you want to observe some live specimens. But let's have a look again under the microscope. Where is it? Yeah, here they are. Let's do the following. Let's go here. Yeah, that's better. Yeah, so the nitrocellulose, this is basically powder that you can buy, and you just mix it with a little bit of water, the nitrocellulose powder, and you allow it to stand overnight, and then it makes this very thick solution. And then you can add it also not only to those brine shrimp, but you can also add them to protozoans and so on to slow them down. Yeah. Yeah, 8 to 24 hours for hatching. Mine took a little bit longer. I think this also has to do a little bit with the temperature, with the ambient temperature. So I had to wait 24 to 36 hours around. Because, again, this is very temperature dependent, as so many things in biology. Yeah. Yeah, you can search for the so-called for sea monkeys, and then if you buy a kid's set, then you're gonna get one which is really contains a lot of plastic dishes and whatnot. But actually, you want to buy them, in a shop for people who are keeping aquariums. Yeah. Can they be kept for longer periods to produce another generation? I think if you are able to adjust the everything, I see no reason why not. Yeah. 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 So okay. Yeah. Let's do the following. I'm going to do something. I'm going to drop a cover glass now on top of them. Okay, maybe I can go up with the magnification this way yet further. Because you see there is no cover glass on here, therefore I can only stay at a low magnification. And let's go here. Yep. So, the scope view, I have to find them again, where they are. Here they are. Very good. Yeah, they're a little bit compressed as well. That's good. So this means that I can go really high up with the magnification. I think this one over here, because it's larger, it's not able to move quite as much. Let's go up a bit further. That's pretty nice. I like it. I like it always if you're able to look a little bit into the intestine, digestive system. Yeah. So let's go up yet further. I don't know if this actually works. Yep, that's now 40 times. That's almost a little bit too much, I would say. I wonder if this is the pouch for the yolk, for the food. Oh yeah, and that's basically quite nice. So let's have a look if we're able to see the other one. If I find it, here it is. Yeah, this is definitely much younger. Is there anything else that can be used instead of nitrocellulose powder to slow them down? That's the question. Not that I know of. There is in the United States. There is, I think it's called Carolina Biologicals. It's a school supplies company. They're selling a liquid called Proto-Slow. Proto from protozoan and slow, SLO, without the W at the end. Methyl cellulose. What did I say? Did I say nitrocellulose? It's methyl cellulose. Oh my gosh. Of course. Nitrocellulose? Nitrocellulose, isn't this an explosive? Gee. Okay. Methyl cellulose. Okay. Yeah. So nitrocellulose, I think, is dangerous. No, I wanted to say methyl cellulose, of course. Gee, my bad. Yeah, and I can show you just a second. I can try to show you the powder. If I find it. So here it is. It basically comes in a box like this, right? And if you open it, then it's basically nothing more than a white powder, right? And you just basically suspend a little bit of this in some water. And the advantage of this is that it's not osmotically active. This means that it doesn't change the water balance of the organisms. Yeah, it's just very viscous, but it does not impact their osmotic balance. For example, glycerol is also very viscous, but has the real problem that it throws off the osmotic balance of the organisms. Yeah. Does cooling the slide and sample also slow them? Would not surprise me if it does, but I think the problem that we have is by cooling it, you need a special stage where you can adjust the temperature. Normally, there are some stages that can be warmed up, but I don't know about if you can cool them down so easily, because problem is that it's at the microscope being at room temperature is difficult to keep the temperature stable. Yeah. Yeah. So I just got here. What did I miss? Okay, well, at the beginning of the video, I did a little bit of a more or less successful DNA extraction. And yeah, and right now we're looking at a few so-called Artemia brine shrimp, which are essentially also known as sea monkeys, yeah, and sold for kids as well. And I've added now some methyl cellulose to slow them down. Here we go. Yeah, yeah. Otherwise, what I would then try to do next is the following. Now that we have more or less enjoyed those little critters here, I would actually move on, and I would like to, before I throw out the plant, maybe I would like to try to make a cross section of the stem again. So basically, this is a follow-up of what I've done last week. I need to just clear everything away because otherwise I'm kind of worried that, yeah, I'm knocking something over. So let's put everything away here. So, and I need a sheet of tissue paper. And what I would like to try to do now is the following. I would like to try to make a cross section of this stem here, which I put into a little bit of a stain. And because I would like to see how this looks like. Okay. So maybe or maybe not it works, I don't know. So the idea is as follows. Yeah, I put some stain in here. You don't see it here. Here it is, and maybe some of the stain was actually drawn up into the plant. I don't know, but I would like to see that. So I need to make a thin cross section, and I don't know where I put my sharp razor blade. Where is it? Just a second. Here it is. Okay. So I'm going to do it like this. Okay. I don't know if maybe you're able to see that maybe here on the side a little bit, it looks a little bit bluish. So what I would like to do first is I put this away and I put this here under the stereo microscope. We're going to have a look under the stereo microscope. Maybe it's able to stand. Here we go. Yeah, I was hoping that there were more of them. Yeah. You see that there are also blue spots in here. These are vascular tissue where some of the stain was actually drawn up into the plant. Yeah. For whatever reason it did not quite reach the others here, but at least you see that on the inside here, yeah, these are blue spots. Yeah. So let's try to do the following. I would like to try to now make a cut of this. And in order to do that, I'm going to take out again my microtome. And this is a little bit, maybe a little too much, so I'm going to cut it again here. So I try to put it in here. So other way. I need a slide. So, and I'm going to now again make a cut here to remove this part first. This is the first one here. Is a little bit the most difficult one. So, and then now I can start to advance it out a little bit. I hope I'm turning this now into the correct direction. So let's have a look here at the first one. Okay, this was a little too little. This one was already a bit better, and then what I'm going to now do is I'm just going to place all of them directly on here. Okay, next one. Here we go. This one looks quite okay. Let's try another one. Okay, here we go. And the whole thing goes under the microscope, and we're going to now have a look. You know what? I might add a little bit of water. And you see that these are up over here. You see that there are these tiny blue dots here, and I would like to see maybe these are the places where the stain was drawn upwards. So I need now a little bit of water, which I don't have. Where is it? Over there it is. So, and a pipette. A little bit of drop here, a drop here. Cover glass goes on top. On top here, and also on top here. And maybe a little bit more. Yeah, the sections are a little bit thick, but maybe we're able to see something. So let's have a look. Again, here we go. And the scope view, it's very thick, of course. Yep, okay. Yeah, here we see some of the blue spots here. This is actually what I wanted to demonstrate. Maybe there are nicer ones over here. Yeah, here we go. This kind of illustrates, or is kind of a little bit the evidence that these are the vascular bundles. So these are basically the cells that transport water up to the plant. And because I put the flower into the stain, you can see that it's those that actually start to stain first. Because the stain, in this case the crystal violet, was pulled up through those vascular bundles over here. That's why they started to stain first. Yeah? The others didn't stain, and I think the reason could be maybe if the vessels are closed at the bottom of the plant, right? Because maybe it was cut off not properly, and if water is not able to enter, then it's of course also not able to transport them. Yeah. So, but essentially, you see over here that it's the vascular bundles that start to stain first. Yeah. Let's do the following. Let's have a look over here. If this looks, yeah, here as well. And this is so-called xylem. The others did not stain here, but over here again, yeah, it's stained. You know, it's always around the, yeah, over here as well. And what my hope was actually with the flower, but the flower was already a little bit too old, is if you actually take a flower and you put it into some ink or some stain, what's going to happen is that after a couple of hours, the flower itself will also start to turn blue because the stain is drawn up and then goes into the petals, and then you can also change the color of the flower there. right? This was a little bit the hope that is going to happen, but the flower was already a little too old. But at least we could see that the stain actually went up a little bit down here at the stem. Yeah. So let's have a look here. Some questions. How long was it soaked in the stem? I left it in there now about, I would say, 10 hours. But it, as you can see, it did not work very well. Yeah. Have you at the cross section of a grafted plant? No, I did not do that. So I did water it down a little bit, indeed, because the actual crystal violet would have been way too intensive and and it's too strong. That's the whole point, because the stain, if it's too strong, it indeed might kill the cells and damage the plant, and that's why I added only several drops of the stain to some water to dilute it down. Yeah, indeed, in order to prevent the plant from being completely killed. Yeah. So I'm just checking some of the questions here. Yeah. Can you show the pistil under the microscope? The pistil. The pistil. Okay. That's basically this central part here. Let's put it first under the stereo microscope. Where is the stereo view? Here. Where is this? Yeah. You know what? This doesn't work well. So what I'm going to do now is I'm just going to cut the plant, and so it's easier. Yeah, it's easier to have a look at. So here we go. This is the female part of the flower, which receives the pollen. Okay. And I can see that there is already some pollen on here. And what's going to happen is I mean, this is actually something that I wanted to explain when the pollen germination would have worked, which it didn't. The pollen that will fall on here, they will start to grow, germinate, and it will grow a pollen tube all the way down to the ovary. The ovary of the plant is down here. Okay, it's got to be down here somewhere. And this is basically where the egg cells are, and the ova, and this is where the fertilization happens. Yeah? And the pollen tubes, the growing pollen tubes, is actually something that you should be able to observe quite nicely under the microscope. However, you gotta get the sugar concentration right. You have to add some other elements to stimulate the growth, and unfortunately, it didn't work. Okay, so I'm working on it, and in springtime, I hope that when there are more plants, I can try more plants. As a matter of fact, yeah. So this is the thing. I would like to do the following. There are a couple of questions here which I can quickly answer. Can I stain this with aquadine orange for more contrast? For heaven's sake, don't. Aquadine orange is highly cancer causing. Okay, so aquadine orange is indeed used for staining DNA, but when we worked with aquadine orange in the lab, we had to put on double gloves. Okay, so because any stain that actually messes with the DNA will might do DNA damage and therefore causes cancer. So with aquadine orange, you have to be really careful. Yeah. So aquadine orange, that is also used in gel electrophoresis when you want to make DNA visible with UV light, but that is something where you have to be really careful. Okay. And I'm not even sure whether you can actually get it that easily. Yeah. So I'm just reading here. Most of the questions I already answered. Okay. Yeah, nitrocellulose. No, not methyl cellulose. Again, my bad. Yeah, it's methyl cellulose powder. And I think this methyl cellulose powder is also used it's called special ingredients, I think, for food. When you want to make food a little bit thicker, then you can use this, for soups or whatever, right? So the plant I put away again, and before, for the last, well, 5 minutes or so, before I call it a day, I just have just a few organizational things that I would like to mention. In beginning, when is this? Maybe is it in 2 weeks or so? There might not next week is not a problem, but in 2 weeks I think I will not be able to do a live stream. I will also announce this in the community post because I'm not here. I have to go somewhere over the weekend, just for your information, and a few other things. And I need a little bit the help or the feedback of the community, of the microscopy community. When I make videos, not only on this channel but also on the other one, the main channel, I do some microbiology explainer videos. It's sometimes difficult for me to think of topics because so if you have any recommendations of any not microscopy related but any microbiology or whatever topics, please, I welcome any feedback concerning that and any suggestions on topics, because it makes makes it a little bit easier for me then to meet the expectations of the community. Okay, so if you've checked my other channel, the Microbehunter channel, the main one, you'll have seen that over the last couple of months I've done quite a few. I've shifted the topics away from microscopy. Microscopy is now on this channel here, and I've shifted away in the other channel from microscopy to more towards the impact of microorganisms on daily lives, our daily life, so to say. And if you have any specific requests, I welcome them. Okay, so that's one thing. Another thing that I want to inform you is yes, while I do have already quite a few newsletter subscribers, I do admit that I have not been sending out many newsletters recently. However, I would like to start doing that. I'm kind of reworking the whole plan a little bit on how I keep in contact with the community. I've been experimenting around also with different formats and so on. I just wanted to tell you that there is a newsletter as well, and I will be sending out maybe some additional comments or ideas relating to the live streams, and just to keep you a little bit updated. What else do I have here? I think otherwise, pistol and okay. I think that's it. What I'm going to do now is I'm just going to call it already a day, because I'm already a little bit tired, as you probably can hear on my voice. I hope that you found it nevertheless interesting, even though the whole banana thing did not quite work the way that I hoped it would work. But in any case, now you can have now you have at least a method where you can try to experiment at home. Okay. I'm going to call it a day, and I wish you all the best. Okay. Happy microbe hunting. That's the standard greeting. Okay, and I hope to see you next time again. Bye bye.